we're going to start with what we did last week, or last time, which is there we go. Good. Okay. Which is this assignment, or this activity we were working on. So a single slide of just this is in that PowerPoint. If you guys would get yourselves into your groups, let's see. Whatever. I mean, that would be fine. You can break up into different groups if you want. It doesn't matter. You're not being graded. You're just talking. Break yourselves up into groups of 3. We may have different people here. Some people were absent last time, so just whatever groups. And one of the groups can, in fact, 4 of the groups can connect to this individually if they want to that screen. I don't know if you want to do that, but let me show you what this slide is that I have here. So if you click on one of the pictures, oops, let me see. Let me actually turn it on. If you click on one of these pictures, it will open up for you a window of that image that you can then manipulate and look at, as well as having the model. So if you want that asset as you're doing this activity, please open that up. So here is, you guys get beta D glucose, you get something, a mess, a sugary mess, beta fructose, oh how sweet it is. And what do you get? Oh yeah, ribose, the first enzymatic protein, or enzymatic molecule. You guys get beta galactose over here, you're with them. And you 3 get that. And then you 2, what do you get? Okay. There we go. Yes, the heart of it all, some deoxyribose. So I'm also going to hand out these little tools that I had last time, and you can use the pair of these, which are chiral enantiomers, if you remember what that word means, non-superimposable mirror images of one another. Oh, somebody switched these around. Don't do that. They're meant to be non-superimposable mirror images. So mirror images that cannot be superimposed onto one another. Okay, that's what makes something chiral. Mirror images that are non-superimposable. So these represent the definition of what that means. And you can map these onto each of the atomic centers. For example, here, this atom, is this chiral or not? Well, I can map this onto it. The blue is going to represent, well, let's just do it this way. The blue is going to represent everything hanging off this bond. Everything hanging off this bond. It's blue. We're just going to make it all one little blue ball. This red thing here is going to be this little green ball. This carbonyl group is the silver ball, and the white one here is that. Okay, so we've mapped that. Now, can I take this and superimpose that on that same mapping? If I can't, then this is chiral. Okay? If I can, then it is not chiral. You will see what I mean. There are going to be chiral atoms and non-chiral atoms in your molecules. All right? I want you to appreciate what this really means. It's hard to get your heads wrapped around three-dimensional space, even though you've been swimming in it since the beginning. Here, I want a group. Oh, you're over there. Yeah, I want a group of 3. There's that, and then here's this tool. All right. Yep. So your goal here, last time you guys verified or didn't the CH2O ratio, and I wanted you to determine what N was if it did pass CH2O. If it didn't, I want you to look at the molecule and try to figure out why. Where did it go off the rails? And then the third thing is, how many chiral centers are there? All right? I'll mix about and help you. How's it going here?
Pulling up the slideshow.
Yeah.
So, it matches C6H12O2. Like it is 612-6 as well. Mm-hmm. So we just have to figure out if it's chiral.
How many chiral centers, if any, are there? Yeah. It's the chirality of sugars. The chirality of sugars is one of the things that it's going to be important to appreciate when we talk about what sugars actually do. So I want you to understand what I'm actually talking about when I talk about it.
I went to the farm yesterday.
Yeah.
Really cool place.
It's amazing.
Nice. Ben wasn't there.
Oh, it's too bad.
Because I had emailed him the day before. He's like, yeah, I'm going to be there all day.
He's a busy dude.
I went, and then I ended up talking to this guy, Cliff.
Okay.
For a little while. I mentioned he said he didn't know you were, but we're just one of
a host of dudes there, yeah.
And so I talked to him, but I'm gonna go back today to try and catch Ben because Ben is supposed to be there too.
Cool.
So
Yeah.
After lab.
The dude, I should say this to the whole class. That due date that I put up there is not like a hard due date for today. It was a timeline. By next weekend, I want the draft.
Okay.
Yeah. So that's okay.
Yes, that'll be. And I really just need to interview Ben.
Yeah.
Because there were definitely questions that this guy Cliff was super helpful for, but then there were other ones where he was just honestly, you would need to ask.
Yeah, I know. I wish I had thought to get in touch with you sooner that weekend they were having the rice thing, because they had a whole crew of people out there trimming the hemp plants. And Ben was there. It would have been a good day for it. But yeah, it's cool. Are they still trimming out there?
Yeah. The first crop they had to take in because it couldn't withstand a hard freeze.
Oh yeah.
A hard frost. But then they still have the two other ones
hardier
that are able to survive hard frosts, and they were. I think their concerns were quelled because there's already been 2 or 3 hard frosts this year, and they're all the plants are really, really healthy still. So they're really excited about that.
They're pretty protected down there. It's weird land because it's sort of in a valley. It's kind of swampy, which is why he was able to get it so cheap. But it might have protected them a little bit from that. I don't know.
Are we emailing you links to SoundCloud? Is that how you want?
Yeah, that's for next week, right?
Okay.
Yeah, today I just want okay. I'll address that to the class.
Cool.
Yeah. How are you guys doing?
How do you actually tell people?
How do you do what?
How do you tell? I don't know.
Okay. So let's look at this carbon, shall we? So let's just line it up like this, so that actually, how is this going to go? Like this, all right? So that white is white, silver is this hydroxyl group, the green is the ring oxygen, everything hanging out in this direction, like this carbon looking in that direction, and this carbon looking in that direction is purple, all right? So, all right, we've got it mapped. Each of the 4 unique substituents around here is uniquely represented by something different. Okay, so then let's take this one. So we said that the hydrogen was the hydrogen, right? And we said that the ring oxygen was the green, all right? So we did that. But we said that the silver was the hydroxyl group, yeah? And the purple was this black. So okay, let's fix that. Let's make it so that, what do we want here? We want the silver to be the hydroxyl and the purple to be the ring carbon. Well, that works. I got that fixed, but now the other 2 have been switched. So the green is, okay, so I can't map this one onto this one as well. So this must be a chiral carbon. It's a chiral carbon. 4 totally different substituents in the tetrahedron. Let's now look at this carbon, for example.
It's just carbons, right? Nothing else.
Actually, you can have other molecules that are chiral, but hydrogens, yeah, oxygens are not going to be chiral because there's only 2 substituents on them. Yeah. And if, for example, I were to, well, we won't get into that. So let's just look at the carbon. All right, so let's map this. Let's have this white equaling a white and a green equaling a white, okay? And the purple equaling a red, this hydroxyl group, and the silver equaling the black carbon, okay? So, good. We have white, a white is a green, purple is a red, and a silver is a black. Let's get that mapping going. Okay. So now I have the silver is a black, like I had before, and I had a purple is the hydroxyl group, as I had before. And I also have a white mapped onto a white and a white mapped onto a green. It's not chiral. I can map both of them onto the same thing. And that's essentially because these 2 hydrogens are identical. So when the carbon looks in that direction, it sees the same thing as when it looks in that direction. You get it? Understand chirality? So go ahead, figure out how many chiral centers are there. Yeah, hey Damon.
So one clarifying question.
Please.
When you say hanging off part, like which part from the center or each center is considered?
Here, hand it to me. So I'll show you how to map it here. You have, in this one, let's have the hydrogen, center to center, hydrogen mapped onto a hydrogen. Let's have the silver represent a hydroxyl group. This brown is going to represent this ring oxygen, and this green is going to represent the attached ring carbon right there. That's our mapping, all right? So let's set it down. Hand me the next one. All right. So let's get the hydrogen mapped onto the hydrogen, the center to center. The silver was the hydroxyl group, right? But this isn't oriented quite right, is it? Because the green is onto the red.
And it was this one.
Yeah.
Right.
So let's fix that. Let's fix that. Okay, so now the brown is on the ring oxygen and the green is back on the carbon. But
it's not.
Those 2 are switched,
Right.
okay? So we can't superimpose both chiral enantiomers onto this carbon. That means this must be chiral itself.
Yeah.
So let's look at another carbon. Let's look at this one here, for example. Okay, so let's map it like this. So carbon is on carbon, hydrogen is on hydrogen. This green one is going to represent that hydrogen, and hydroxyl to the brown and the silver to the black there. Okay, so let's set that down. We remember that that's our mapping. So let's line it up with the silver on the black and the brown on the hydroxyl, and the white goes to a hydrogen, and this green goes to a hydrogen as well. So we have mapped both chiral enantiomers onto the same carbon, which just means that this carbon isn't chiral. And that's essentially because these 2 substituents, these 2 things hanging off that central carbon, are the same. They're the same thing. So when this carbon is looking in that direction, it sees the same thing as if it were looking in that direction. So you can superimpose the mirror image of this carbon onto itself, and it would get the same thing. Does that make sense?
Yeah.
Chirality, life in 3 dimensions. Yeah, so go.
Confused about what do you define as the part that's.
So here's a carbon. It has 4 bonds coming out of it. Does it see something different in each of those 4 directions?
So for this direction, would it only be this part or this whole part?
It's whatever you see hanging out. Like when it looks, imagine being this atom. When it look down the bond. When you look in that bond down that direction, what do you see? I see a hydrogen sticking out that way, a hydroxyl out this way, and a carbon sticking out that way. Okay. It is attached to all this stuff back here, but when I look down this way, I see something different. I see a hydroxyl sticking out that way, or a hydrogen, a hydroxyl there, a ring oxygen down there. It's just seeing a different thing sticking out that way. So it's this carbon would be chiral. It's non-superimposable onto its mirror image. Make sense? Yeah. Okay. Is everybody divined the deeper paradox of life in 3 dimension here? Okay. So let's just go around. Why don't you guys share what you found? Who's first? Beta glucose, you're first in everything in life. Why don't we make you go first?
Cool. So we decided that there were 3. So yeah. Oh yeah. 3 Chiral Centers because we went by. So imagining this is like a black bead. It's like that. One Chiral Center.
That is chiral, you're saying?
Yes. Yes. Chiral. As that one and that one as well.
All right.
And we were
So let me pull up glucose here. I hope this is the open chain form. Oh yeah, good. Thank God. All right. So you guys are saying this carbon here is chiral. This one is chiral, and this one is chiral. You say N is 3. Not N, I'm sorry. You say N is 6, but you're 3 chiral centers. What do you guys have to say about it? Let's consider this one first. Let's look at that. Does that carbon in the center see 4 unique things looking down each of those bonds? So let's look down this bond from this carbon back here to this carbon. We look down this bond and we see a hydroxyl, a hydrogen, and this mess of a sugar chain hanging out there. Okay, so those are all 3. That's a pretty unique pattern. Then let's look down this bond. If I look down this bond, that carbon sees an oxygen hiding back there, which is attached to a hydrogen, a hydroxyl group. So that's definitely different than the other bond we looked at. Okay. Now let's look at the third bond, which is this hydrogen-carbon bond. If we look down that bond, what do I see? I see nothing but a hydrogen down there. And so that is indeed different to the other 2 bonds that we've looked at. So let's look at the fourth one, this carbon-hydrogen bond. We look down this bond, and what do I see? I see a hydrogen. I see actually exactly the same as what I saw down this bond. That suggests what to you, Annika? What do you think? Yeah, that carbon is actually not chiral. It's not chiral. So if you were to take the little tetrahedrons that I gave you guys and map those onto each of the 4 substituents, you could map both of those models onto that substituent without there being a conflict. Okay? It's a tricky thing. It is a tricky thing. You have to get in there and hold these things, get your head wrapped around it. So you said this was a chiral carbon. Why'd you say this was chiral? Don't be shy. I just blasted the biggest hole in your boat that I'm going to. You guys are on good ground.
Off, like, if this end, like, these are both carbons, then this blue would be the next carbon. Is that enough bonds?
Yeah.
Unless of like looking down the bonds.
Mm-hmm. Well, I like to look down the bonds because that helps you put yourself in the perspective of the carbon and really understand what the carbon is seeing. So let's look. When this carbon is looking down this bond, it sees a hydroxyl. Looks down this bond, it sees a hydrogen. Looks down this bond, it sees 2 carbon fragment, each with a hydroxyl on it, and it looks down this fragment, and it sees a 3 carbon fragment, 2 of which have hydroxyls, and the other of which is this carbonyl group up here, this acetyl group. So all 4 of those substituents are different, and that is a chiral carbon. You were right. Yeah, you got that one right. And you identified this one as being chiral as well. So let's look at this. We have a hydroxyl, a hydrogen, a big long nasty carbon chain, and an acetyl group. So also correct. Good job. Does anyone have any other comments about this molecule? What do you think? Your brow furrowed, Peg. You're thinking hard. What is Dr. K getting at? What am I supposed to be seeing?
Trying to think of what it could be, but I was like,
Yeah. Well, they gave me some good stuff, but they didn't give me enough of a good thing. Keep going. Let's look at this carbon. They considered this one, and this one along the carbon chain. What about this one? We have a hydrogen, a hydroxyl, 2-carbon fragment, and a 3-carbon fragment. All 4 of those substituents are different. All 4 are different. This carbon is chiral, as is this one here. So in fact, there's 1, 2, 3, 4 in a row. We identified this one as not being chiral because these two things are different, or are the same. These hydrogens are the same. And let's look at this carbon. So this carbon is planar. It's planar. If you look down the carbon-carbon bond, you'll notice that that carbon that's attached is in the same plane as the oxygen and the hydrogen. It's planar. It's not tetrahedral. All 3 of these, in fact, all 4 of these atoms are in the same plane. And so because of that, there is no chirality. You could take the mirror image of this carbon and superimpose it on itself. So this carbon looks the same from this side as it does from this side. All right, it's a non-super. It's a superimposable mirror image. It's not, so only these tetrahedral carbons become chiral. All right, so this is not a chiral carbon. You didn't make that mistake. You didn't call it chiral, but that was a tricky one. That was the one. The 2 things in all of these molecules that are the most tricky are what's called the hydroxy methyl group, this little tail that's sticking out most of your rings. And then these guys had the extra problem of having an open chain and having this carbon, which in the closed ring is in fact chiral, but when it's open chain, it's not. This is what's called the anomeric carbon. We talked about the anomeric center last time. It doesn't have a name, so it doesn't change the name, meaning this is glucose. This is still glucose here. That carbon that's got the double bond O right there, when it's in the open chain or reducing form, it's not chiral, and it's not actually changing the chemical nature of that molecule. All right, that's why it's one of the reasons they call it the anomeric center. It's not chiral in the open chain form, but becomes chiral in the closed chain when the ring closes. So here would be this anomeric carbon open chain. Here it is closed chain, and when it's in the closed, even though this is fructose, that doesn't matter. It has 4 different substituents around that tetrahedral carbon. All right. In chemical terminology, it goes from an sp2 hybridized to an sp3 hybridized carbon, which is just a way to describe its geometry. All right, who's next? What do you guys got? What atom?
Last side we're beta fructose.
Oh, beta fructose. Yeah, this one. Cool. Let's take a look at it. All right, and you have a pyranose or a furanose? Pyrano is 6-membered. Furano is 5-membered. It's okay. So you have the 6-membered. Nope, that's 5. So look at the ring. Look at.
The ring. Oh, I thought you meant like how many carbons.
No, it's a hexose because there are 6 carbons in it. But is it, when I say pyrano versus furano, if you'll remember back a couple slides to last time when I was talking about the ring shape, is the ring shape a furano, which is a 6-membered ring, or a what did I just say? A pyrano is a 6-membered ring, and a furano is a 5-membered ring. So that's a furano. Fructose can form a pyrano or a furano, and I've given you the furano form there, which is the more common one in nature. but I couldn't find a good model of it online, so I have the Purino form right here. That's okay. We're going to compare the two of them. So what did you guys find?
They were four chiral centers, pretty much the two on the outside aren't for the same reason that they're supposed to be. They both have two columns, the ones on the inside. We couldn't map them onto each other to have like the same, so like two things that have the same. So
4 chiral centers. Yeah, beautiful. Great. Given the lesson that we just learned about here with glucose, that I told you about the C1, the anomeric carbon in glucose, what would you guess when fructose is an open chain? How many chiral centers would be in that chain? What do you think? 3, yeah. Because the anomeric carbon, the carbon that has 2 oxygens attached to it, the one that has a hydroxyl and the ring oxygen attached to it, that would be a keto group. Find the carbon that has 2 oxygens attached to it. Nope, that has 1. Keep going around the ring. It's in the ring. There. See how that carbon has 2 oxygens attached to it?
Oh, yes.
Yeah, in the open chain, that would have only 1 oxygen attached to it, and it would be a keto group, a double bond O group, and it would not be chiral, but it is chiral there in the closed ring. All right? So the chirality of these molecules goes up when they form rings. So you had 4 centers. Let's look at the Furino form. Furino has, this is no, this is yes, 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 yes. So 1, 2, 3, 4. And then this is no, because it has 2 hydrogens attached to it. It's in the 5-membered ring, this carbon is sticking out as a tail, but in this 6-membered ring, it's got incorporated into the ring. And that just is a measure of this anomeric carbon that in the open chain form has a double bond to an oxygen, whether it's going to react to carbon 5 or carbon 4, or I'm sorry, carbon 6 or carbon 5 when it forms the ring with that hydroxyl group on either of those carbons. Am I freaking you guys out? Are you all right with this? I know this is some real chemistry for people who haven't had organic chemistry before, but you're doing okay. It's really just a toy. These are just toys. We're understanding what these models do in space, but they do represent the sugars that we eat. All right, Annika, you guys. What were you guys on?
Ribose.
Oh yeah. Okay, ribose. And that's a Purino or Furino? Furino. It's the 5-membered. Yeah, this is the one. I couldn't. Yeah, right. I have the Purino form on the model, but I couldn't get the Furino form. Okay, so what did you find?
I got 3, and then I went through again, and I got 0.
Okay. Okay. So which 3 did you get? Okay. So 2 out of the 3 is right. The one that you're holding has the 2 hydrogen is wrong because that has, when the carbon is looking down, either of those hydrogen bonds, the CH bonds, they're the same. It's seeing the same thing in both directions, right? So it's not, each of the 4 points of the pyramid are not unique. 2 of the points are identical. But there are 3. The 3, so there's a 5-membered ring. One of the members of the ring has 2 substituents, 2 hydrogens on it that are of course identical, and that's not going to be chiral, but the other 3 are. The other 3 are. All right. Let's see if that holds in the Purino form. So what do we have here? We've got 1, 2, 3, 4 here. And yours is 3, right? Yeah, hold it up. Let me make sure. Let me look at it. Yeah, 1, 2, 3. No, 4. There's 4 there. There's 4 there. Yeah, okay. So if you look at the 4 carbons in that ring, oh, that's ribose. Yeah. Sorry, I'm having my mind is having a hard time with it this morning. There are 4 carbons in that, and each of the carbons in that ring has 4 unique substituents around them, meaning that each of them is chiral. Okay, you see that? So it's 4, and we have this one is not chiral. Hydrogen and hydrogen are the same, so we can turn this around and sort of look at it here. These all 4 of these are not unique, whereas the 4 substituents around here are unique. Heading in this direction is a different thing than heading in this direction. Likewise with this carbon, likewise with this one, and then the anomeric carbon is certainly chiral. All right, what about it, Damon et al.? What'd you guys find?
So the first time we went there, I got 4 questions.
You guys have galactose. Yeah, beta galactose. Can I ask you? Oh, okay, my computer's frozen. Here we go. Can I ask you if it confirmed the CH2O? It did. Okay, and what was your N? I forgot to ask you guys, what was your N? Did yours confirm, so that's ribose, did it confirm CH2O? And what was the N? 5. So that's a pentose. You both had hexoses. Your N was 6. This N was 5. Your N was 6. So we had 3 hexoses, 1 pentose so far. And all of you have confirmed CH2O thus far. All right. So tell me about the chiral centers.
We know that the carbon that's not chiral is the 2 hydrogens.
Okay, good.
And then I'm pretty sure all of the ring carbons are.
Okay, so let's see. Yes.
We didn't really get a chance to talk about it because we were just like figuring out how to do it the whole time.
Okay, I see. This one here is, those are all 4 different. All 4 of these are different because going around the ring this direction is a different thing than going around the ring this direction. If I go this way, I hit carbon, carbon, and if I go around this way, I hit carbon, oxygen. So that's different. So that's 1, 2. This carbon now, let's see. All 4 of these are different. And all 4 of these are different. And then lastly, all 4 of these are different. So there are 1, 2, 3, 4, 5 chiral centers in that molecule. 5 chiral centers. So what do we have here? We had 5, 3. What was yours? 4. And you were, what do we say, 4? Yeah, there's 4 there. 5, 3, 4, 4. All right. What about you?
Deoxyribose.
Okay. What is deoxyribose a part of? Do you guys know? We haven't talked about it yet. What is it? Yeah, it's part of DNA. All right. And ribose is part of RNA. Okay, let's hear it.
And you couldn't verify if it's missing.
You could verify this, could not.
Yeah.
What do you mean?
It's doesn't have oxygen.
Okay, so if you're telling me it fails the CH2O test, what is the actual ratio?
There are 5.
Okay, and how many hydrogens? Yeah, so it's C5H10O4. Okay, is that right? I'm asking. Yeah, it is. Yeah, it's deoxyribonucleic acid. It's had one of the oxygens ripped off of it. So it's what's called a deoxy sugar. Deoxy sugars play important roles in a lot of places in biology, but none nearly as important as deoxyribose, 3-deoxyribonucleic acid. It's the carbon 3 that has been deoxygenated. So why don't you let's look at that. Actually, this is a weird molecule of, yeah, look at that. So this is actually not the molecule I gave you. This deoxyribose is deoxy in the wrong position. That's the crystallographic database for you. No problem though. It's the same general principle. So how many chiral carbons did you guys find? 3. So we have, this one is no. We've established why. 2 hydrogens. This one is yes, yes, yes. And then this deoxy site here is a no. It's a no. Yeah, the deoxy site is in a different spot in your ring. That's the way DNA looks. I guess I didn't catch that the structure I pulled up out of the database was deoxy in the wrong spot. But all right, a little bit of exploration into sugar structure. I didn't put my watch on, so you guys keep me on track with time, would you? All right. So just to connect a little, I'm not going to give you guys a quiz on the podcasts this week. It was a short week, and the podcasts were short. I didn't put a lot of. They were both pretty brief this week, under a half hour between the two of them. But one of them, the Fresh Food by Prescription podcast, I did want to touch upon something just to help you put that in context and understand specifically why I chucked it in this unit. What was that podcast about? Yeah, Damon.
Someone, a guy had type 2 diabetes. His hemoglobin A1c was too high.
Good job.
By eating fresh food, he was able to reduce it, and it got rid of a lot of his carbon.
Yeah. So we're going to talk about diabetes a little bit more when we actually get when I do the metabolism with you guys. But I wanted to explain something more fully that was in that podcast, and Damon totally nailed it. There was hemoglobin A1c. What that is, why that's a marker, and why it's related to diabetes. So in general, diabetes mellitus, there's type 1 and type 2 diabetes mellitus. They are characterized by extremely high blood sugar. Okay, for a number of, there can be the two different reasons that you have them. And when you have really high blood sugar chronically in your body, that just means that the glucose levels in the blood are high, and you're not, maybe if you have type 2, you're insulin resistant, or type 1, you're not producing insulin at all. And the insulin is the short peptide hormone we talked about in the protein section a little bit that helps your cells absorb glucose. It turns on the glucose transporter, but it doesn't work. That process is broken in people with diabetes. So their blood sugar is really high all the time, unless it's under control. One of the things that happens is these amino acids that are on the surface of your hemoglobin, which is the molecule that's in your red blood cells that transports oxygen from the lungs to your tissues. The positively charged or basic amino acids that are sticking off of them are these three here. There's lysine, arginine, and histidine. all these nitrogen-containing amino acids that are positively charged. They're sticking off these NH2 groups, these amine groups, are sticking off the hemoglobin molecule. And when there's a ton of glucose, we've already seen that glucose can go from open to closed chain form, right? It interconverts. And even though only 0.25% of the glucose in your blood is open chain, when you have super chronically high glucose, that amounts to actually a significant amount of glucose. So when it's in the open chain, that double bond O that is on the end of the chain of the glucose can react in a reaction that we're going to explore in lab at the end of the semester. It's the same reaction that gives rise to the delicious smells that we have when things cook. But it is the reaction between a protein and a sugar. Here, it's pathologic. So we have this glucose molecule bonds, that aldehyde group reacts with the amine off the lysine, histidine, or arginine, and it's basically a dehydration reaction. So a water molecule is released, and you'll notice that there's an oxygen and 2 hydrogens that are now missing from that equation in the shift base. And then it goes through what's called an Amadori rearrangement. Again, this is part of the Maillard reaction we'll explore when we bake cookies at the end of the year. We'll understand how that relates to like the smells of food. But here's some pathology related to the same chemistry that glucose goes through. So it gives you this hemoglobin, and it's called Hemoglobin A1c. A1c is just a label that is in reference to how this hemoglobin was first isolated, how it was first isolated on what's called column chromatography. That's not important. But it's a type of what's called glycated hemoglobin. Glycation is a major problem for people with diabetes, who have chronically high diabetes. And hemoglobin is just one of the, I already told you there's between 100 and 140,000 different types of proteins that your body makes. All of them are susceptible to glycation if they're exposed to chronically high levels of glucose. And so hemoglobin A1c is a marker for diabetes. You can test the blood, and it's very reliable test for the level of what your blood glucose has been just over the last 3 months, because hemoglobin has a lifespan of only about 3 months, 90 days. And so if you have a lot of hemoglobin A1c, that's going to tell the doctor over the broad arc of 3 months, have you had high blood sugar or lower blood sugar? So it's a nice medium term. It's not like the daily pinprick that diabetics have to take to see what their moment-to-moment blood sugar is. It's a medium-term way of tracking blood sugar in the body. Hemoglobin A1c is, when you have it glycated like this and many other proteins, they become dysfunctional. They don't work the way they're supposed to. They cause inflammation. It leads to atherosclerosis, or hardening of the arteries, arterial plaque, and it basically gums up your capillaries and leads to peripheral neuropathy and a lot, like it can be hearing loss, impotence, a lot of unfun problems from having high blood sugar. And so this diet, this fresh fruit by prescription thing, which was a really short clip, it was only a 6-minute clip, and it didn't go deep into the science. But essentially what's happening here is when you're eating food that has a lower what's called glycemic index, that means that it's going to liberate the sugar in the food into your bloodstream more slowly. It's going to liberate the sugar more slowly into the bloodstream. This is related to the types of carbohydrates you're eating. How much fiber, soluble and insoluble fiber. We're going to be talking about soluble and insoluble fiber next week in class here, and how that can relate to blood sugar. All right, so that's just a little explanation of what they were talking about and how that relates to carbohydrates and glucose. Are there any questions about that?
Yes.
Yeah, what's up?
So, like, with your hemoglobin A1c,
what hemoglobin does, or A1C?
Yeah, the A1C part.
Yeah. Hemoglobin A1c there's two features to it. Number one, it is a marker for medium-term high blood sugar. Okay, if you have elevated levels of A1c, that means you've had chronically elevated levels of glucose in the blood. And it is also directly pathologic in that it can lead to inflammatory cascades in the body that can be the precursor to the hardening of the arteries and the clogging of peripheral blood vessels that have a lot of consequences like hearing loss, vision loss, impotence, peripheral neuropathy, ulcers, gangrene, all of the unfun aspects of being diabetic or old or both.
Are there other classifications for
Yeah, so A1C is a description of hemoglobin that, as I said, the label describes how it was isolated. And there are other, when you're taking a sample of whole blood that has a lot of hemoglobin in it and you elute it across a column, so you're going to have a column that is packed with some kind of solid phase, meaning like stationary phase material that's not moving, and then you're going to run a fluid or a mobile phase through the column, and whatever you're eluting, your protein here, is going to dissolve in that solvent, and you're going to have differential interactions between the types of different proteins that are dissolved. There's going to be a differential interaction with the stationary phase as it goes through the column. And you're collecting fractions. You're going to fill a test tube, label it, set it aside. Collect another test tube, label it, set it aside. A1C refers to the elution profile of hemoglobin on a certain type of column. But there's going to be a range of fractions that you're going to be isolating in hemoglobin. And A1C is a group of hemoglobins that have a certain level of glycation. So there's going to be hemoglobin that comes through that's not glycated at all. There's going to be a fraction of hemoglobin that maybe has one glycation site that comes through in a different fraction. And then maybe you'll have a fraction that has 2 or 3 in it, and then a fraction that has maybe 5 or 6. And then you'll have a fraction that's A1C that's got 10 or whatever on it, right? So this is just like a category of pathologic hemoglobin due to the glycation that it has because of elevated glucose levels. Does that make sense?
So
I mean,
A1C has 10 glycation sites?
I actually don't know that. I don't know that. That was me just explaining how it works. Yeah. I mean, as you guys get older, your medicine is heading towards preventative medicine, and all of you guys are probably going to be having, you may already do it, have annual checkups to go to the doctor. And as you get older, they'll certainly do blood draws on you. They may do it now. You get an annual blood draw. As you head into midlife, like my age, they'll be looking at hemoglobin A1c. There'll be a blood test that's done to you guys in the future. So this is what it is. You can tell the nurse about it when she's taking the blood out of your arm. All right. Any questions on that? How much time do I got? Oh, okay. There you go. Wow, 2 slides.